cells develop initially in the primary lymphoid organ, which is the bone marrow, and then it moves to the secondary lymphoid organs, lymph nodes, spleen, and mucosa associated lymphoid tissue. In the bone marrow, they undergo stages of maturity from the hematopoietic stem cell in the absence of Notch1 signaling into progenitor B cell, pre-B cell, and immature B cell. Now, all these stages are antigen independent maturation because they are happening without the help of the antigen. So, any of the tumors that arise at this stage is called as leukemia, and it happens in the bone marrow or in the blood. So, all of these are precursor B cell stages, and hence all of them are going to be positive for CD19, which is the pan B cell marker. Now look at the stages. The progenitor B cell is going to be positive for CD19, and that is the pan B cell marker, and for other immaturity markers, that is your CD34, HLA-DR, and TDT. Now this will undergo some immunoglobulin heavy chain rearrangement into pre-B cell, then followed by immature B cell. So pre-B cell is going to have CD10, and another important cytoplasmic heavy mu chain, which is further going to form immunoglobulin M. So next is going to be immature B cell stage, which will now acquire CD20, and CD10 is going to disappear. Along with that, it will now have surface immunoglobulin IgM. From this immaturity stage, it is going to move to the peripheral blood, and this immature B cell will undergo class switching and somatic hypermutation to form the activated B cell, but that is still naive. It has not yet been exposed to the antigen and is not activated. So that naive B cell will remain in the peripheral blood and is going to be positive for CD23, and CD5 may or may not be positive. From the blood, these naive B cells are going to enter into the lymph node via what? Via arterioles, and there they are going to encounter what? They will encounter the antigen, which is being presented by the dendritic cell or the macrophages, coming via afferent lymphatics. So naive B cells here, when they are going to be activated by the presence of the antigen, this is known as antigen dependent maturation. So the lymph node, if you see the structure wise, it is going to have a capsule, a subcapsular sinus where the macrophages reside, a cortex, and a medulla. So if the B cells are not activated, it is going to form a follicular or the primary follicular area. As you can see, the lymph node has an interfollicular, follicular, and the perifollicular area. So once activated, this follicle is going, interfollicular area has naive B cells. The follicular area also has naive B cells being formed. So the primary follicle is composed mainly of these mental cells or naive B cells which are not activated, and they are small lymphocyte with cleaved nucleus. But once the activation is going to happen, they are going to form a germinal center. Now this germinal center will then have what? It will have centroblast in the darker areas and centrocytes in the lighter areas. So all these cells of the follicular area are going to be positive for BCL6 and CD10, and apart from these centroblasts and centrocytes, they will have follicular dendritic cells and T follicular helper cells, which are going to help them in T dependent activation. So this germinal center is said to be a polarized area. Why polarization? Because we had talked about that it has some darker areas and the lighter areas. Now the centrocytes are now then going to mature to form short-lived memory cell and the longer-lived cells or the plasma cells. Now these memory cells reside in the marginal zone and they are positive for CD27. Since they are short-lived, they are going to produce only one type of immunoglobulin, that is IgM. But the plasma cells are the longer-lived cells and they are going to produce antibodies of all the types IgG, M, A, E, and they are then positive for CD38, CD138, CD79, but negative for CD19 and surface immunoglobulin. The normal kappa lambda ratio that they are going to produce when they are a normal plasma cell is always 2:1. But in all the lymphomas, you will have some sort of restriction that either one type of a light chain is being produced more. So that kappa lambda ratio changes. So it is not just in plasma cell myeloma, on any kind of lymphoma, the skewing of kappa lambda ratio can be present. So from this, we can now understand what all lymphomas arise from, what all cells or what all areas of the lymph node. Now the plasma cell, as we talked about, is negative for CD19, negative for surface immunoglobulin. Why? Because it itself going to produce antibodies. So why will it show immunoglobulins on its surface? So the lymphomas arising from the interfollicular area are the mantle cell lymphoma. Because these mantle cells are present in the interfollicular area. And why? They have an immunophenotype which is different from the normal naive B cells. So the mantle cell lymphoma cells are positive for CD5 but negative for CD23. Now the lymphomas which are arising from the follicle are follicular lymphoma, DLBCL or diffuse large B-cell lymphoma, Hodgkin's lymphoma and Burkitt's lymphoma. And all these tumors are going to be positive for germinal center markers which are BCL6 and CD10, and they are going to be negative for CD5 and CD23. Now the lymphomas arising from the post-germinal center area are going to be what? They will be positive for both CD5 as well as CD23, because all the cells which are arising in the perifollicular areas are activated and CD23 is a marker of activation. So you have marginal zone lymphoma, CLL and plasmacytoma arising from the perifollicular area. Please remember, plasma cells are not just going to remain in the lymph node. They will move from the lymph node to reside into their permanent home, that is the bone marrow. This is a table from Robbins to summarize the important CD markers. So please remember, CD10 is positive in the pre-B cells and the germinal center B cell. CD19 is a pan B cell marker which will be present in all but not plasma cell. CD20 arises in the immature B cell stage and it is present in all the cell stages till mature B cells but not plasma cell. CD21 is an EBV receptor and is also present in all the mature B cells and the dendritic cells. CD23 is a marker of activated mature B cells and CD79 is present in conjunction with CD22 in your pre-B cells and mature B cells.